Hi everyone, my name is Tate, and I'm super stoked to be talking to you today about Kleptocnidae in nudibranchs. In this presentation, I'm going to set the stage to start by providing some crucial context, and then I'll delve into some of the unique features that enable some nudibranchs to steal specialized defenses from their prey. I'll discuss how these stolen defenses operate within the nudibranchs and potentially why. And then I'll highlight other examples of this ability across the animal kingdom. So let's just jump right in. Predation avoidance strategies are ubiquitous and exhibit considerable diversity across the animal kingdom. These adaptations may encompass behavioral, morphological, or chemical defenses that arise through a range of evolutionary processes. The prominent way in which cnidarians defend themselves is with characteristic specialized cells known as cnidocytes, which contain sophisticated organelles known as nidae. And these nidae have the ability to inject venom, penetrate tissues, and adhere to their target. The most ubiquitous or omnipresent type of needy is the nematocyst, which is composed of a long thread that is coiled within a pressurized capsule. And so when the cnidocyl apparatus, which is a cilium-like structure situated on the epithelial surface, is mechanically or chemically stimulated, the nematocyst opens up the capsule and the tubule is explosively everted. This is one of the fastest processes in the animal kingdom, actually. And like most defenses in the animal kingdom, nematocysts are generated endogenously by cnidarians. However, some species have evolved a unique ability to acquire functional nematocysts from their prey and store them in their functional form within their own cells. And these acquired nematocysts are referred to as kleptocnidae. Klepto meaning to steal, and nidae is that organelle we've been discussing. And so the ability to sequester entire structures, such as organelles, is pretty rare. And one taxonomic group that's actually capable of this rare phenomenon is the aeolid nudibranchs. Aeolids acquire nematocysts from cnidarians through trophic interactions, predation. And this is associated with some pretty significant challenge. Nematocysts have been documented to effectively deter predators through the physical damage they inflict. And these visualizations here on the right demonstrate that finding. And so aeolids employ strategies and possess multiple attributes that enable safe trans or consumption and transport of nematocysts in their internal tracts. One way in which aeolids overcome cnidarian defense is by strategically targeting nematocysts with lower potency. Likewise, it's been observed that some nudibranchs will actually target immature nematocysts that are incapable of discharging and continue to mature within the organism itself, and most specifically within the cnidosac, which is a structure I'll talk about later on. In addition to protecting their exterior surface, this strategy minimizes the internal damage as nematocysts are transported to the serrata. And the serrata are these kind of extensions of the digestive system, these sacs on the dorsal side of the animal. And one prominent study suggested that the pattern in which some nudibranchs feed induces an acclimation effect. The researchers Conklin and Mariscal in 1977 described a feeding pattern that involves four distinct phases: approach, initial contact, withdrawal, and reapproach. The nudibranch that they studied experienced nematocyst discharge only in the initial phases of feeding. However, when it subsequently reapproached its prey, the nudibranch was able to traverse the oral disc and tentacles of the anemone without eliciting further discharge. Seemingly, initial nematocyst discharge elicited some physiological response that enabled predation. And so this leads me to the next section of the presentation, the physiological adaptations. The exudation of glandular secretions provides a physical layer of protection for aeolids. Mucus is produced both on the external surface of the organism and within their digestive tract. It also impairs the normal sensory triggers for nematocyst discharge. It's believed by some researchers that mucus is secreted in response to nematocyst discharge, thus explaining how the nudibranch in that case study I just mentioned was able to feed when it reapproached. Several anatomical and intracellular structures of nudibranchs are reinforced with chitinous material. This includes the radula, the cuticle of the head alimentary tract, and in these intracellular granules called spindles. The existing literature has emphasized the particular importance of spindles, which are present within most cells of the skin and the stomach epithelia. And upon impact of discharging nematocysts, the muscles beneath the basal lamina contract, causing it to fold and compress the vesiculated epidermal cells, releasing the spindles. And so the impact of this evolutionary novelty of nudibranch skin is multifaceted. Firstly, the spindles absorb the impact of the discharging nematocysts. Secondly, they help to establish distance between the nematocyst and the underlying basal lamina, the muscle layers, and the nerve cells of the organism. And then thirdly, the liberation of spindles causes the meshwork of nematocyst tubules to actually detach. And so another aspect about acquiring Kleptocnidae is that they need to retain the functional attributes of the consumed nematocysts. Therefore, aeolids must minimize the discharge and digestion of these structures as they move through their internal tracts. In addition to the aforementioned mucus discharge inhibition effect, the immune response modifications seem to play a pretty important role in the preservation of nematocyst structural integrity. Some researchers have speculated whether phagocytosis of nematocysts in aeolids is mediated by a microbe-associated molecular pattern and pattern recognition receptors. That's quite the mouthful. But the validity of this postulation is diminished by the observation that nematocysts become separated from their nematocyte, the cell, within aeolids, which means that they don't possess an identifiable membrane-associated antigen. And therefore, Martin in 2003 outlined the existence of a modified cellular mechanism that protects nematocysts from digestive degradation. Typically, phagosomes fuse with lysosomes, thereby exposing the internalized entity to lysosomal hydrolytic enzymes. However, this study found that the nudibranch under investigation had phagosomes with nematocysts that were clearly separated from heterolysosomes. The absence of a phagocytic membrane surrounding nematocysts isolates them from cellular digestive processes that require membrane binding for enzyme activation. And so now I'll discuss how these nematocysts are actually integrated into the physiology of these aeolid nudibranchs. The thing is, the diverse morphology of aeolid taxa means that there are unique mechanistic pathways by which Kleptocnidae are formed. However, we have a pretty good idea of a general framework which is broadly applicable. In this framework, firstly, intact nematocysts pass through the aeolid's digestive diverticula and are transported by ciliary action to a structure known as the cnidosac, where they're engulfed by cnidophages. For clarity, in this review, the term Kleptocnidae refers to nematocysts after they have been sequestered in these cnidosacs. And you can see them in these figures here of this aeolid as well as in these diagrams. And so, thirdly, the Kleptocnidae can actually remain within the cnidosac for days to weeks and may even receive metabolic support from the organism. And then lastly, as new nematocysts are incorporated into the nidosac, older nidophages and the Kleptocnidae are pushed distally towards the tip of that serrata. Recall the serrata are these kind of sacs on the dorsal side of the organism. And so to actually release the Kleptocnidae from their serrata, they follow this process where, firstly, sensory bundles of non-motile cilia near the tips of the serrata are directly connected with nerve cells that coordinate the contraction of complex musculature around and of the nidosac. And this in turn forces the extrusion of the kleptonidae from the cytoplasm of the nidophage. And secondly, the orientation of microvilli funnels the kleptonidae towards the tip of the serrata. And when the Kleptocnidae reach the external environment, it's been observed that they discharge from their capsule. But why? Why are they doing all this? Why have they evolved this elaborate mechanism? Well, a lot of researchers think it's for defense, and this is reinforced by one study that saw that several potential predators of nudibranchs fed more quickly on aolids without their serrata than the ones that had intact serrata. Um, but some researchers are trepid to accept the defensive role of Kleptonidae with absolute certainty because it's difficult to distinguish between the defensive role of Kleptonidae and other chemical defenses that are present in some species. And this has led some researchers to believe that the nidosac may just function as an excretory organ. But I think it's important to consider that the function of Kleptonidae doesn't need to be mutually exclusive. It could be both. Now I just want to highlight that there are organisms with Kleptonidae across the animal kingdom in different phylums, including the Ctenophora, the Coelomorpha, and Platyhelminthes. And so here are some pictures of nematocysts within each of those groups. And I think it's really interesting that there are several shared characteristics among these groups. And I think that these help rationalize the independent arise of Kleptonidae in these groups. And so, firstly, they all have soft bodies without a hard exterior. And therefore, I think that this encourages some alternative mode of protection to evolve. Secondly, many nematocyst sequestering species have branched or diverticulated digestive systems. And then thirdly, many have the ability to regenerate tissues, which is important considering their limited ability to control nematocyst discharge. There are also similarities in the way that nematocysts are sequestered across these different groups. So firstly, many species transport nematocysts from their gut or near their gut to their storage location using nidophages. And secondly, there seems to be some selective sequestration of particular nematocyst types in multiple groups. And then finally, in both nudibranchs and platyhelminthes, the sequestered nematocysts are stored in those muscular nidosacs I discussed. And so if I had to summarize, I'd say that nudibranchs are pretty cool. But seriously, uh, I hope you've learned in this presentation that cnidarians have nematocysts, which are endogenously created specialized defenses. Aelid nudibranchs can steal nematocysts and store functional forms of them within their cells. And the aelid nudibranchs have a plethora of protective attributes that enable them to do this safely while retaining the functionality of nematocysts. I also discussed that Kleptonidae may be used for defense or excretion, or maybe both. And then I highlighted examples of Kleptonidae in other taxonomic groups, which indicates convergent evolution. Thanks everyone for listening. I hope you enjoyed.